Hey Beakling, so today what we're going to do is draw an overview of cellular respiration. And I already shared these papers with you. If you need them, you can shoot me an email. But as you can see here, we're going to fill out a whole bunch of stuff today. Okay, we probably won't cover this today. We'll do this in a later video, or I'll do this with you, fermentation. But today we're going to cover the first 3 pages of this. All right, so these are the colors that I'm going to use today. You can use whatever you want, to be honest with you, but just try and keep it about the same. All right, so I have like a purpley pink, okay, and then just common normal colors here. All right, well, let's get started. So the first step is really knowing what the equation is for cellular respiration. All right, and that equation is glucose, right? C6H12O6 plus oxygen. Okay, and I'm going to balance it already because I know the equation. So 6O2, and that's going to give us waste products, 6CO2. These are our products, right? These are the reactants. These are going to be the products. Plus 6H2O. Plus, the whole point is energy, right? And in this case, the energy that we get is ATP. And we also lose some as heat. Okay, so what is it about this equation? Well, first of all, what do we do in this chapter? We're taking glucose and we're going to steal the electrons from it. And that's going to give us CO2. And remember what that's called? Oxidation, right? When you take somebody's electrons, you oxidized it. Now, where do those electrons eventually end up is on water. So oxygen will accept the electrons with some hydrogen ions or protons to form water, and that's called reduction. And by the end of today's pictures, you should really be able to see what that's all about. Okay? Now, the goal is really this chapter to get ATP, right? We are very happy to get some energy there, so we love our ATP. Unfortunately, thanks to the second law of thermodynamics, anytime we're doing these energy conversions, we're also going to release useless energy for us, right, in the form of heat. Okay, so unusable energy gets released. So we're going to take our glucose molecule, and as long as we're breathing, we'll be able to do cellular respiration. What do? We steal the electrons from glucose. That's called oxidation. And eventually that leaves us with CO2. What do we do with those electrons? Well, at the bottom of an electron transport chain, oxygen is going to grab the electrons and some hydrogen to become water. What was the whole point of this? And what we'll see today is how we do it. Well, to get energy. Okay, and we get energy in the form of ATP, which is what we're excited about, and heat. Now, I'm acting like heat is a bad thing because we lose so much of it, which it's not great. However, we've evolved to do that now. So imagine if you were born and you weren't a constant body temperature of around 98.6 degrees. Well, that's how your body's set up. All your enzymes work that way. We would die if we weren't losing all this heat, right? So now that we're set up to do cellular respiration the way that we do it, that's just what it is. All right, let's move ahead. All right, so our next step is called glycolysis, and glycolysis, if you want to think of it that way, sugar breaking. All right, and we're always going to start with a molecule of glucose. All right, and I like to underline my reactants and my products and things like that. I'll put them in green so they stick out a little bit, so I know what goes in and what comes out. Now, you're well aware that glucose is C6H12O6, right? That's the molecular formula. I'm not going to draw all the H's and O's. What I'm going to do is I'm going to draw 1, 2, 3, 4, 5, 6. So all I'm drawing here are the carbons. Okay, I'm not going to draw all the H's and O's, but you have to remember that they're in there because we're going to be manipulating them, okay, throughout the process. So there's my glucose molecule. The next thing I want to remember is, where does this happen? Well, this is ubiquitous, meaning it can happen in just about any organism on the planet, right? And that's because everything has cytosol or cytoplasm. So this glycolysis occurs in cytoplasm. And I'll put a red box around that to remind me that, oh yeah, okay, that's one of those things where I try to remember where all these processes happen. All right. And I think we're ready to begin. So the first step, here we are in this chapter, we're trying to get energy out. And the first step, we actually have to put energy in. So what we're going to do here is we're actually going to take 2 ATP, all right? And I'm shortening this up a whole lot. There's a lot of enzyme catalyzed steps here. We're just going to overly simplify today. But you're going to take 2 ATP, right? Now let's make it like a starburst or an energy burst usually when I'm doing ATP. And we're going to go ahead and phosphorylate this molecule and thus be left with 2 ADP. Okay, so 2 ATP come in here and they lose their phosphate groups, and that leaves us with ADP. Sure. All right. And what that did was we now have this molecule here that is phosphorylated. Okay, so there's my 6 carbons. But now, thanks to ATP, I added a phosphate group to each end. Okay, and I'll color them to make them stand out a little bit. Okay. Now, because we added that energy, okay, that is what's known as the energy investment phase. Okay, and there's different ways to say this, but energy investment phase. All right, so we're trying to get energy this chapter, and here we go. We just started by not getting energy, but using energy, right? All right. So now that we added the energy, what can happen is we can break bonds in glucose. Okay, now, again, this is overly simplified because don't forget, glucose is actually a ring in aqueous solutions, right? So I'm drawing this like a straight chain, but it's obviously a ring. But what happens is we can now break a bond. So the fact that you broke this bond, something awesome this chapter can happen. Because we're in cellular respiration, anytime we break a bond, there's going to be cool stuff that happens. Okay. So what actually happens is there is this cool dude sitting there, and if you remember, he's an electron carrier. Okay. You have a coenzyme. His name is NAD+. NAD+. Okay. And NAD+ is hanging out near there. Okay, and I'm trying to draw it the way your book shows it with 2 little notches in the top. And what NAD+ says is, oh boy, did you just break a bond? Well, how about you reduce me? Okay, I'm going to, in other words, NAD+ is oxidizing the molecule and stealing the electrons. Remember, this is the redox reaction. And I'll draw them blue because next chapter this will make a lot of sense why I'm drawing these electrons in blue. And what these are high energy electrons. Okay, very, very important step. Now, when that happens and you steal the electrons, okay, or you oxidize the molecule, what can happen also is not only did you steal the electrons, but you also ripped off hydrogens to do that. So I'm just going to draw a little H+ here showing that you also ripped off hydrogens. And the reason that happens is because this is a dehydrogenase enzyme. Okay, so let me get caught up. I'm going to fill in the other side with the same thing, and then we'll talk about dehydrogenase. Okay, so as you can see here, all I did was I drew the same thing happening over here, okay, because it happens to both of these molecules. Now, what exactly is this dehydrogenase enzyme? Okay, and I'll draw a little side portion here, and I make this stick out as well, because this is very important for your understanding of how this chapter really works. Okay, so we have this enzyme, and it doesn't really matter how you draw it. I'll just draw it like this, sure, something like this, and then maybe I'll go like that. Okay. This enzyme's name, it's a whole family of enzymes that do this. I'm going to call him dehydrogenase. Okay. So, one second, I'll color them up. Okay. So, next, and you could of course pause the video as we're going if you need to color or anything like that. What's going to happen is this little intermediate, meaning something between glucose and where we end up, they're all called intermediates. So this intermediate right there happens to fit into the active site of this dehydrogenase enzyme. So I have a carbon, a carbon, a carbon, and a phosphate group, right? All right. The other thing that's going to fit in here, if you remember, NAD plus is a coenzyme. So technically, this little guy can fit over here. Okay, so there's my NAD plus. So see, what's actually happening here is this dehydrogenase enzyme, okay, came and tried to grab the hydrogen off of here. The problem is it doesn't work on its own without a coenzyme. So that's where NAD plus comes in. NAD plus also fits into the active site in this case, okay? And what happens is NAD plus says, yeah, sure, here, let me grab your electrons, okay? Thus, NAD plus gets reduced to form NADH, and as dehydrogenase rips off the hydrogen, you release some of them, right? Some of them. One of them gets carried by NAD plus to become NADH, and one of them gets released. Okay, so remember, going back up here for a second, we only drew C6. I did not draw the H12O6. Okay, but that H12 and O6, they're in here too. So we just lost some hydrogen thanks to dehydrogenase. Okay, these enzymes come in there, rip off the hydrogens. And what do they do when that happens? Well, they're always going to have this little coenzyme, little electron carrier with them. Okay, and as they rip off the electrons, they reduce the electron carrier, NADH, NAD plus becomes NADH, and they also release protons, right? Remember, hydrogen, that atom only has one electron, okay, and one proton. It doesn't even have a neutron. So what happens is when you steal the electron, it's just a proton, okay, and that's what this H plus is here. And that happens, of course, from both sides. So it happened over here as well. We released a proton, we took another proton because NAD plus with one negative electron would be NAD, and then when we add the other electron, it would be NAD minus. Oh, so add the proton, NADH. Okay, there you go. Okay, so this dehydrogenase enzyme happened over here as well. So I'm not going to keep drawing that, but for the rest of our drawings, whenever you see yourself filling up some electron carriers, whether it be NAD plus gets reduced to form NADH, or later in our drawings, FAD becomes FADH2, then you know that dehydrogenase was hard at work. Now, because we're keeping this pretty simple, I'm not really going to get into this step much with you, but this guy now grabs another phosphorus. It's during this enzyme catalyzed reaction that you can actually get another P group added on, but I don't want you to think of this other P group as another ATP that we're spending. You're actually using the energy available because of the reaction to have this inorganic phosphate added. Inorganic phosphate just means a phosphate that's not connected to an organic molecule. So this P, in other words, a lot of my students will say, wait, how did you get that P? And it's a great question. Okay, but for right now, because we're really simplifying this, you don't have to worry about it. You just have to know that it's not coming from necessarily ATP. Okay, and again, if you care, a lot of times this thing right here, when you see it in a book, it'll say something like P sub I. Okay, and that just means inorganic phosphate. Okay, so this next step is very, very important. We took some electrons out, okay, and these are going to be very important later, okay? But now we have this molecule here, and what we're going to do is, all right, we're going to take the phosphorus back off, the P group. And how we do that is, well, here comes our buddy, ADP, and we're actually going to create ATP. Cool. And we do this on both sides. ADP becomes ATP. All right. And what that's going to do is that's going to leave us with a very similar molecule as what we had. Okay, so they are different though. I know some of you might be saying to yourself right now, wait a minute, isn't that where we just were? No, I'm drawing it that way, but there's little differences between these molecules here. Okay, so this molecule, in other words, looks just like this molecule. Okay, but they are different. So ADP became ATP. So we just made 2 ATP. Hey, we're finally getting some energy out. All right, cool, cool. All right, in our next reaction, what happens is we actually lose a tiny bit of water. So just a little bit of a waste product from both sides. H2O. H2O. All right. And I'll rearrange it a little bit just so you can see that is changed again. But again, these things are pretty much the same thing, not too different. Whoop. All right. So we lost some H2O from both sides now. Okay. And now here comes another important step. You can see I magically colored in the water there. Okay. Here we're left with just the 3 carbons, and of course there's hydrogen and oxygen in there that I'm not drawing. So wait a minute, if we lost another P, well, what's that mean? That means ADP came in again and ATP from both sides. All right, so now we got even more ATP out, and we're left with this gorgeous molecule called pyruvate. Okay, or you can also call it pyruvic acid. Okay, one is ionized, pyruvate. And because that's like our main end product of glycolysis, I'm going to go ahead and underline that in green so I don't forget about it. Okay, pyruvate, very, very important. Now, a couple more things to talk about here. First of all, how are we getting this ATP? A main part of this chapter is going to be talking about oxidative phosphorylation. That is not what we're doing here, okay? That's going to be something later that we have to learn about in these drawings. So all that happens here is this is something getting your ATP, all right? So I'm going to draw a little arrow again, because this is like a sidebar here to make sure that we understand exactly what's happening. All right. And this is something that we call substrate, so you know we're talking about enzymes, right? Substrate level phosphorylation. Okay, so substrate level phosphorylation, all that really means is, again, we're going to have an enzyme, and let's say if it's this intermediate in it. All right, so here we go. We have this enzyme right here. And in this case, we have all different enzymes depending on what step you're on, but in this step, we're right here. We're going to have that guy coming in here, and we're going to have ADP coming in here, right? So give me a second, I'll color this up. Okay, so as you can see here, what happened is we have an enzyme. Okay, now in this case, it's this guy right here. And what's going to happen with him is he's going to take in this intermediate. All right, so we have an intermediate here. And we want to get this phosphorus off of there, right? We want to make some ATP. Well, we're going to have an enzyme that's going to say, okay, I'm made specifically for this enzyme or for this intermediate right here to fit in my active site. And then I'm also going to put ADP in my active site. And what happens is you transfer the phosphate group. The enzyme lowers the energy of activation. Remember the E sub A, okay, that energy barrier, so that a phosphate group can get connected to it. And then what are you going to release from this enzyme? Well, you're going to release this now intermediate, right? And you're going to make ATP. Okay, and of course ADP would then get released as well. Okay, oh, I'm sorry, no, the ADP is ATP. All right, so ADP gets released as ATP. This intermediate, which by the way, these all have names. Okay, they're all named. We're not going to have to worry about that. That also gets released, and now you can continue. So far we've talked about just major broad categories of enzymes that happen here. We talked about dehydrogenase enzymes that go in there and rip off hydrogens, okay, and that allows us to take the electrons or to oxidize whatever intermediate so we could reduce the electron carrier, NAD plus in this case. And we also are learning about, to make ATP, you might need enzymes during these steps to work, and that's via substrate level phosphorylation, okay. And through this whole thing, we're finally at the bottom, and we have pyruvate. Now, the whole point of the bottom of this is really we paid energy up here, so technically we lost 2 ATP to start. But now we gained, let's go ahead and count them. 1, 2, 3, 4. Okay, so what we have down here now, not only the ATP, but also the high energy electrons, this is called the energy payoff phase. All right, so energy payoff phase. All right, and those are very important steps there. So energy investment phase and the energy payoff phase. Okay, pretty neat. And now I always like to talk about our totals here. So give me 1 second, we'll do that. Okay, so totals. All right, a little hard to see there, it's all glary. All right, for our totals, can you think about how many ATP did we get altogether? Right, and if you said 4, that's good, right? 1, 2, 3, 4. However, we lost 2. So how can we say that we lost 2 and gained 4? You could just say 2 net. Okay, so 2 net ATP. That's important. Okay, and that word net is very important. What else did we get as far as like very important products here? NADH. Those electrons are going to be really important, especially in our last picture. So we got 2 NADH. Very, very important. Water was created as a byproduct, so I just write that down here. Sure, water. But one thing that I really want to make sure that you all understand is we also lost, thanks to the second law of thermodynamics, heat. Right? And I write it all goofy on here to remind myself that, yeah, that's that one thing that we lost there that we can't use. As a matter of fact, when I draw, a lot of times on the board, what I'll do is I'll take my marker and I'll go psh, 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 and I put all these red lines coming out of there to remind kids that, yeah, as we're going through this, we definitely lost heat, right? And that heat technically increased entropy, and we're not going to be able to use that energy. So by us breaking down and then eventually getting the ATP from our food, we're actually losing in the long run, okay? But we'll talk about that later. So there's our first picture. There is glycolysis, all right? And what glycolysis does is it starts the oxidation of glucose in the cytoplasm. Again, very important to remember, we did not draw the H12O6. We're just drawing carbon, but they're in there, okay? This happens in the cytoplasm. First thing is, you put in 2 ATP, that busts apart the molecule. That's called the energy investment phase. So we had to spend energy this chapter early on so we can make energy overall, right? Now that you broke it apart, okay, you can see you're phosphorylated. That provided the energy to break the bonds of glucose. Now you can actually go ahead and reduce NAD plus to form NADH. And how does this happen? Well, there's these family of enzymes called dehydrogenases, and they're very, very, very important. And dehydrogenase will come in here and grab whatever intermediate, okay, depending on which intermediate, that depends on which dehydrogenase will fit it, right? And then NAD plus fits in there, gets reduced, and now you have high energy electron carriers. When you get good at this stuff, you know that these are more than likely on their way to the mitochondrion, right? And now what happens is, well, another phosphorus shows up. Just don't forget, this inorganic phosphate does not come from an ATP. And now you're going to get your ATPs back out. Yes, you lose a little bit of water as a waste product, but now we're grabbing these 4 ATP. Okay, we're very happy. Now, how do we get these 4 ATP? Well, substrate level phosphorylation is how you make ATP this chapter, unless you're at the last step, okay, which is called oxidative phosphorylation. So in substrate level phosphorylation, you just have an enzyme grab an intermediate and takes a phosphate off and puts it onto an ADP, right? And that's how we make ATP. So what was our overall totals from the energy investment phase and the energy payoff phase of glycolysis? Well, we got 2 net ATP, we got 2 NADH, we got some waste products, sure, and definitely we lost some heat along the way. Okay, let's go ahead and start our next picture. Actually, before we start that next picture, I do have one last question for you. If somebody asked you right now, hey, you started off with glucose, which you know is an energy-packed carbohydrate, right? It's a little monosaccharide. And the reason it has so much energy is because of the carbon-hydrogen bonds, which we're messing with, right? By the time you're done glycolysis, where is most of the energy of glucose? that used to exist right here. Where is it now? Here's some answers that students have given. Well, it's in the high energy electrons in NADH. Good, but no, that's not where most of it is. Some kids will say, oh, it's in ATP that we collected. Good. No, that's not most of the energy, right? Then some kids will say, oh, the heat, we lost a whole bunch of heat. Excellent. But no, that's not where most of the energy is. If you really think about it, you really just split a bond and lost a little bit of stuff as you're going. Pyruvate. Pyruvate is where most of the energy of your glucose is right now. Okay? So what we need to do is we need to get to the next picture. Okay? We need to get what organelle do you think we're heading to, because we need to get a whole lot more energy out of our organic fuel here. And you know we're heading to the mitochondrion. And the reason, partly, that I like to think in the back of my head, why do we do glycolysis? Well, you had to make your pyruvate because pyruvate is welcome into the mitochondrion, whereas glucose is not. Okay, so the fact that you made your huge glucose sugar into smaller pyruvates or pyruvic acid, now we're ready to roll. Okay. Okay, so this one is not too difficult to wrap your head around. We're just going to do the grooming or the oxidation of pyruvate. Now, before we can really start here, it's really important to understand where you're at. Okay, so for example, this phase occurs in the mitochondrial matrix. Okay, so I always like to know where I'm at. Okay, we were just in the cytoplasm and we just made pyruvate, right? But now that pyruvate is going to enter into the mitochondrion. And if you don't remember what a mitochondrion is, what I'm going to do is I'm going to draw one over here, okay, because it's super important in this chapter to know where you're at. Mitochondrion. All right, and if you remember, a mitochondrion has two membranes in it. You have the outer membrane, and then you have this inner membrane, which folds and stuff are called cristae. So what I'm going to do is I'm going to go ahead and color and label this, and then I'll hold it up to the camera so you can see and pause the video if you need to label it. All right, so here we go. Here you can see I have the mitochondrion, right? And in the mitochondria, okay, or mitochondrion, this internal part, this is the fluid-filled part, this is called the matrix, okay? Then the outside is the outer membrane, and the inside, the internal membrane, we call that the inner membrane. Okay, remember all the folds and stuff we called cristae. We were learning about cells. And then the space that I colored red here, that's also very important in between, is called the intermembrane space. Okay, meaning the space in between the matrix and the outside. All right, so remember, this thing probably used to be a prokaryotic cell, right? There's actually DNA in there, which is cool. It's extranuclear DNA that we have. And we used to think we got all of the DNA here from our moms, but now new research says that, oh, your dad also might have given you some mitochondria when the zygote formed. Pretty cool. Okay, so where we're at right now is we're there where that X is. We're inside the matrix. Okay, so here comes pyruvate. Remember, that's where we ended up in our last picture. So pyruvate is how we're going to start this. And this is a pretty quick picture here. All right, and what's going to happen is I'm just going to combine all of this all at once here. We're going to end up going through a couple changes and ended up with somebody down here. Now, the first thing that happens here is we break a bond. We lose a carbon. Okay, now before we didn't lose any carbon, we just rearranged bonds and we lost a couple little atoms here and there. But point is, now we're actually losing a chunk of our molecule here. Okay, and whenever you break a bond and lose a carbon in this chapter, we can remember that you're actually losing CO2. Okay, so the fact that CO2 is released, we now drastically change this pyruvate. And who do you think was sitting there, okay, with an enzyme? Because again, who does this is dehydrogenase. So somebody has to help a dehydrogenase enzyme here oxidize pyruvate. Okay, and that helper guy is known as, I use my same colors as before, NAD+. NAD+ is just sitting over here waiting, and when that bond breaks, he could not be happier because he becomes NADH, right? Pyruvate gets oxidized, NAD plus gets reduced to form NADH. And now we have these high energy electrons right there. We are very happy. Okay. Now, there's a new guy that shows up. Okay, and it's important to have your vitamin B, because one of the things that we do is we make somebody named coenzyme A. Okay, so this coenzyme A shows up here, and he's going to be super duper important. All right, so coenzyme A shows up, and he's going to say, oh boy, okay, I see what happens here. Pyruvate, you just got oxidized, you lost a carbon, somebody's stealing your electrons, and some hydrogen ions, don't forget, so we also released that, okay? And what you're left with down here is, well, the 2 carbons that are left, plus now you have coenzyme A, which we can break it down a little shorter here and just call it CoA. Okay, so the coenzyme guy is the same thing, to shorten it up. And what you call this guy here is acetyl coenzyme A, acetyl-CoA. And he's very important. Okay, now let's talk about our totals, and I'll tell you why this step is super important. All right, so that was an easy part, right? Here's our totals. We got, well, what do you think? What did we get out of here? What do we love that we got out of here is certainly NADH, right? Now, here's a trick question. How many NADH did we actually get here? If you said 1, good thinking. However, if we're talking about the oxidation of glucose this chapter, I only drew 1 pyruvate. And let's not forget that this actually happens twice per glucose, right? So don't forget that. Oh yeah, that's right. This step actually doesn't just happen once, it happens twice per glucose. So how many NADH did we get is actually 2 NADH, right? We got 1 here, and then this would have to happen again with the other pyruvate. We'd get another one. Sure. Now again, yeah, CO2, right? We lost 2 CO2 as we did that, so we lost some waste product. And of course, yeah, heat. Can hear my dog barking outside. Beautiful day. Chasing the birds, probably. All right. So, yes, of course, don't forget we indeed did lose heat. So, again, I would probably draw, if I was on the board at school, a whole bunch of heat coming off of here, just to remind us that as you do these energy or these conversions, you're going to lose some energy along the way. Okay, now there's something very important about this step. Let me explain. So here's the deal. All we really did here was get a ticket to the big dance that's about to go on. What the main goal of this chapter is stealing the electrons from glucose so that we can feed them into this chain that's coming up. The problem is, to get most of the electrons out, we need to enter this thing called the Citric Acid Cycle, or the Krebs Cycle. Well, the problem is, glucose wasn't even allowed into the mitochondrion. So we had to go ahead and make pyruvate first, and thank you glycolysis for that. And we got a little energy out of there, we're happy. But now the problem is, now that we're in the mitochondrion, pyruvate isn't really welcome to the big party that's going on known as the citric acid or the Krebs cycle. So what we had to do is we had to go ahead and groom it into acetyl-CoA. Now, this coenzyme, if you want to think about it, maybe this is like the prom or a dance at school or something. Coenzyme A is like a limousine driver. And he says, hey there, acetyl group. Hey there, you 2 little carbons. How about and don't forget there's other stuff here too, I bring you to this cool dance that's about to happen known as the citric acid cycle, right? And coenzyme A knows just how to get you in there. Okay? So one of the main reasons we had to do this grooming, are we happy about getting 2 NADH? Of course we are. Yeah, very much so. We didn't get any ATP here. We did lose some carbon dioxide, and we definitely lost heat. But now that you're the acetyl-CoA, now we're ready to rock. Let's get into the dance. So where's the majority of energy of my glucose right now? Well, you still have a lot here. Don't forget, there's 2 molecules of this acetyl-CoA. So maybe over here in the totals, in the last picture, I could have also written, hey, we got 2 pyruvate here. And don't forget here, we also have 2 acetyl-CoA. All right, because that's where most of the energy is. But we're going to go ahead and continue with our acetyl-CoA in just a second. All right, so let's have our limousine driver bring us 2 kids to the dance and see what happens. Okay, so where are we at? It's always important to know, right? This also occurs in the matrix. And newer research says it's probably right along the inner membrane, but it's on the matrix side. So I'll say occurs in matrix. I'll just write near inner membrane. Okay. And that'll be important as well for our next picture. So it's always good to know where you're at. Okay. Now, here's where it gets a little technical. Okay. And it's not important to memorize all these little complex words and things like that. But I find that if you just know a little bit about the background without going too deep, it actually helps the overall understanding. Okay, so what we're going to do here is we're going to start with a guy who has 4 carbons. Okay, and we're about to draw a big cycle here. Okay, and what his name actually is oxaloacetate. Okay, and like I said, he is a 4-carbon compound, so I just wrote 4C there. And he's so important that I'm actually going to underline him in green. All right. Now, we just had the acetyl group getting made, right? The acetyl-CoA. So what's going to happen is that's going to be up here. Okay, the acetyl group is floating around inside the matrix. And you know that this guy has coenzyme A attached to it still. All right, so CoA. So this guy is my acetyl-CoA, right? Okay, I'm going to underline him as well because he's kind of the start of this whole thing. All right, now this gets a little weird because here's what happens. As oxaloacetate is nearby, coenzyme A obviously helps the enzyme work this step. Coenzyme A says, oh, hello there, oxaloacetate, who's already in the dance. Okay, I want to introduce you to somebody. So what happens is coenzyme A introduces this little acetyl group, which is 2 carbons. Okay, then coenzyme A says, hi, hey, here you go, kids. This is Mr. Oxaloacetate. Okay, and you don't know if he's a good guy or not yet. Let's wait and see what happens in this dance. And then coenzyme A gets out of there. Okay, he leaves the party quickly. And he'll probably go pick up some more kids and bring them to the dance. Or what they think is a dance. Dun, dun, dun. Oh, there could be some drama here. Let's find out. There's always drama at high school dances, isn't there? Okay, here we go. When you combine this 4-carbon oxaloacetate to a 2-carbon acetyl group, okay, thanks to coenzyme A introducing you, dropping you off here, you're going to create, get ready for math skills, 2-carbon plus 4-carbon would be, you got it, 6-carbon. Okay, and this guy's name is citric acid, or citrate. Okay. So he has 6 carbons. So let's see what happens so far. We have the acetyl group that we groomed up here, right? So our acetyl-CoA comes in here. Coenzyme A says, all right, you kids, here you go. I want you to meet this guy named oxaloacetate. He's a 4-carbon compound. And together, you guys combine to form citrate, or citric acid, a 6-carbon compound. And now, what you thought was going to be what a nice, memorable night is about to turn gruesome. Oh, no. Why is this so gruesome? Well, here we go. This is a little scary. You thought you signed up to draw some cellular respiration, and all of a sudden it turns into a horror film. Okay, so the first thing that happens here is your new little molecule where you guys are stuck in here, right? The 2 carbons of the acetyl group are within here. One of you gets shredded and cut loose. Oh no, we just lost CO2, right? So it's like a big chunk of your dance partner, right? Your date to the dance just got ripped off and goes away. And, well, you know what's going to happen, though. We are in chapter 6 here, folks. So if you rip off a carbon, you know who's very much happy about that, or who helped with that. Actually, with the intermediate being left here is dehydrogenase swoops in. Dehydrogenase says, well, hello there, right? And he's going to rip off some hydrogen. And as long as he has his little helper, NAD plus around, all will be well. So NAD plus is here, and he's going to get reduced as we oxidize, okay, the molecule to become NADH. And you know that he has now received the high energy electrons as he stripped off hydrogen. He also took a proton in, right? So now he's NADH, and the other proton just released. Oh boy, how scary is that? So now we're down to a 1, 2, 3, 4, 5 carbon compound, right? And don't forget, these all have names. All the intermediates we're going to draw have names, but it's not important except to know where do you end and begin, okay? And then you'll see the scariness. So right now, goodbye to one of your carbons, right? Got shredded, and now you are scared. You're thinking, what happened? I got in this, the limousine from this coenzyme A drove us here, and I should have known the limousine was all tinted windows and had some weird fur on the walls on the inside. There was weird stuff happening here. And now I get delivered to this dance, and whoosh, already my partner's gone. So now what happens here? Well, let's see. Next step. Uh-oh. Goodbye, CO2. Now you have also been pretty much slaughtered, right? The CO2 is gone. Now, of course, we're in chapter 6 here, folks, right? In the cellular respiration. So NAD plus, okay, is going to help the dehydrogenase, thus becoming NADH, right? We broke a bond, so you know what that means. We're doing redox reactions, right? Oxidation reduction. We got some high energy electrons. Yep, we released a proton there. Now something else happens actually at this step, which is interesting. We actually have this inorganic phosphate floating around. Okay, where's my yellow over here? So we have ADP coming in here, and we make some ATP. So that's cool. Okay, so ADP plus a little inorganic phosphate, right, is going to create ATP. And if you care, when you get deeper into this stuff, you actually make guanosine triphosphate here, which gets converted to adenosine diphosphate. But so ADP plus P is going to make ATP. And again, this is via substrate level phosphorylation. There's an enzyme that helped us do this and fill this up. Okay? All right. So what's going to happen here is this. Okay, a little different here because you're not taking the phosphorus, again, from a molecule, though, don't forget. Right? This ATP that's created just kind of happens. All right? You don't have to grab an intermediate here. You're kind of using the energy of it. Oh, that's confusing. Okay, so now we're left with a 1, 2, 3, 4. A 4 carbon. And to be honest, there's not much left to you at this point. Right? You've already been kind of slaughtered and taken away in our horror story here. You thought it was a dance, and it turns out it's just this vicious oxidation cycle. It's like the oven of your body, right? Imagine all the heat coming off of here as these molecules are just getting sliced and diced. So what's going to happen from this step is you're going to go from a 4 carbon to a 4 carbon. Oh yeah, but don't forget there's other atoms in here that we're rearranging and changing around, things like double bonds becoming single bonds, etc. Okay, and what's going to happen here is we have a new dude showing up to the scene. Okay, now don't get scared, no big deal, but because we're rearranging bonds, we have this guy named FAD. All right, and FAD becomes FADH2. Oh, baby. Okay, so don't be worried. It's pretty much the exact same thing. Okay, it's just a different little coenzyme here helping out this step. And like I said, even though you didn't break a bond, you did. You're just rearranging. You're breaking and making different kinds of bonds. And that's what opened the door to steal these electrons or oxidize it. Okay, so No big deal here. Just remember it's a different electron carrier, but they are now cleaning up the scene here. They're getting rid of any little remnants that you were ever even brought to this dance. And now what happens is we're going back to oxaloacetate. But in this step, again, you're rearranging bonds. And our friend NAD plus is there to finish cleaning up the mess. And he's going to get reduced to form NADH. Oh my goodness. And look what happens now. We're right back to oxaloacetate. And guess what? There's another unsuspecting group of dance goers getting picked up in their limo right now by acetyl-CoA, okay, or by the coenzyme A. He picks up the dancers, the acetyl group. He says, hey, come here, I want to bring you somewhere. You're going to have a great time, trust me. And what happens is he introduces you to oxaloacetate, forms citrate, and bam! You get destroyed, right? You get oxidized. The whole point of the citric acid or the Krebs cycle is to really steal those electrons. Steal those electrons, right? So, if you care, there's a little bit of water added here. So, H2O actually comes in here, but that is not important whatsoever. Okay, just giving you a little bit more of the backstory here. But what's happening is this. We now have totals. Okay, so let's talk about them before we go too deep. So, totals, and you can help me out here. How many NADH did I get? 1, 2, 3? Of course not, right? If you're good, you know that it is 6 NADH, right? And that's because, again, this happens twice per original glucose molecule, right? Yeah, don't forget that. Okay, so we got 6 NADH. Now we have a new guy here. We got 2 FADH2. Very cool. Hey, we even got some ATP here. Technically, we got 2 ATP, right? Because don't forget, he came out of this step here, okay? And CO2, of course, is released. Yep, that was us. That used to be our husks. Our bodies are gone now, right? This dance wasn't a dance at all. It was just a way to oxidize us and steal our electrons. And finally, oh boy, speaking of waste, heat, right? This is your furnace, okay? You didn't just lose a little heat here. Oh boy, you lost a ton of heat. So if I was on the board, right, I'd be, oh my gosh, there would be heat escaping all over this picture, right? Because we are a furnace, and look at all of this metabolism that just happened here. Okay, we are burning it up. This is mainly why when you're flexing a muscle, when you're working out and things like that, why does your muscle get hot? Now you can tell all your friends when you're at the gym or whatever, oh, hey, guess what? My mitochondria are going crazy right now through cellular respiration, right? A lot of heat being generated. Okay, so if you really think about it at this point, we are to the point now where, wait a minute, we started with glucose, these 6 carbs. It's pretty much gone. Where did our glucose go? Well, we have a lot of it gone as CO2, and oh yeah, we have electrons floating around still, and we have some hydrogen ions around. What are we going to do now? How are we going to get energy? I thought the whole point of this chapter was to get ATP, right? We really want energy in the form of ATP. So how are we going to get it? Well, here's the deal. Your glucose is gone for the most part, right? But we have a whole bunch of high energy electrons in these electron carriers waiting to go here. So what we're going to do now is we're going to go and bring all these electrons that we've been grabbing to the inner membrane of the mitochondrion. Okay? And that inner membrane is where something beautiful happens. And that beautiful thing is called oxidative phosphorylation, electron transport and chemiosmosis. Here we go. All right, now I was gracious enough to give you an entire page here. Okay, so what we're going to do is we're going to fill up a lot of this page. So I'm going to go ahead and start with drawing the outer membrane. Okay, now to do this takes a little bit of time. I'm just going to draw a bunch of circles here. Okay, and this is going to be the outer part of a phospholipid. You remember what a phospholipid is? It has a, you don't have to draw this, but it has the phosphate group. It's a glycerol with 2 fatty acid chains and then a phosphate group at the top. So it really just looks like that. Okay, it's a head with tails, and if you remember, the head is hydrophilic and the tails are hydrophobic. So what I'm going to do is I'm going to draw a bilayer here. Okay, and then I'm just going to go ahead and fill it in. You can do this very quickly by just drawing a whole bunch of circles and then it's going like this. And that just means, okay, the tails. So I'm going to go ahead and I'm going to draw my top membrane here, and I'll be right back. Okay, so there's my outer membrane of the mitochondria, and so I can write that up here. Outer membrane. Okay, and now what's going to happen is I'm going to have a space here called the intermembrane space. Intermembrane space. Okay, and that's going to be this area right here. And eventually what I'm going to have is that twisty, foldy cristae. I'm going to have my inner membrane, okay, about here. I'm just going to write inner membrane. And then down here in the bottom, this is going to be that big fluidy part to the mitochondria. This is going to be my matrix. Okay. Now, what's going to happen here is we're going to draw another membrane here for the inner membrane. However, this one has some proteins mixed into it. So what I like to do is I like to draw the proteins first. All right, so I'm just going to draw one protein right here. I'm going to draw another protein right here. Okay, and I know this might not make a lot of sense yet until you actually see how it's all working together. So you might want to wait until I draw it all out and then draw yours. Here's another one. I'm going to draw another one up here. Then I'm going to draw another goofy looking one here. And then over here I draw a really goofy looking thing like this. All right. And now what I'm going to do is I'm going to go ahead and fill this in just like up above with a lipid bilayer. Okay, so again, I'm just going to put phospholipids all over the place here, connecting these proteins together. Okay, so I'm going to go ahead and keep doing that, and then you can catch up by pausing the video. Okay, so where are we at here? All right, we drew those proteins inside of this inner membrane, okay, and don't forget what that actually means. Okay, the top of this picture right here, okay, this is the outer membrane, that's just the outside of the mitochondrion. The inner membrane space, okay, that would kind of be like the red area here. Then the inner membrane are all this part right here, all these twists and folds and things like that. And the matrix is inside of here, okay, and the fluid. So that's what we drew so far. And if you notice, I labeled some of these proteins that we drew here, okay, and if you go on to take more bio classes, like if you join my AP bio class or something like that, we'll get more into what's actually going on in here and what these little proteins and cytochromes and things like that are. But you can see here you have protein complex 1, 2, 3, and IV that says there, I know it's hard to see, is 4. And then down here you have ATP synthase. Okay, now in a real mitochondrion, these aren't necessarily always going to be set up perfectly in a row like this or whatever, but one leads to the next as we're going to do a whole lot of oxidation reduction reactions. All right, so that's where we're at so far. I'm going to go ahead and underline this stuff. Just remember, we always like to know where we're at this chapter. If ever you're in this chapter and you don't know where you're at, you're doing something wrong, because it's really important to always know exactly where you're at. Okay, here we go. One of our main guys that we talked about this chapter was an electron carrier named NAD+, right? And he got reduced to form NADH. So he picked up a proton, an H+, picked up 2 high energy electrons. Okay, where did most of the NADH come from? Well, the grooming and the Krebs cycle actually happened right here in the matrix. So a lot of those NADHs are right here ready to roll, and we're about to give them to the electron transport chain. So here we go. Let's take an NADH. So now we're starting with NADH, right? He is in the reduced form. He already has 2 high energy electrons. All right. And what's going to happen is NADH is going to say, hey, electron transport chain, protein complex 1, here you go. It's going to donate its electrons. Thus, it's going to get oxidized, right, and become NAD+. It gave its high energy electrons right to the electron transport chain, or this got oxidized and reduced protein complex 1. You want to think of it that way. Now, don't forget, when you delivered your electrons, you also dropped off your proton, right? H plus got released in this case. All right. Now, before I go a whole lot further here on why that's so important, I'll just go ahead and tell you this too. At the second one here, that's where that other guy comes in. Okay, so remember we had FADH2. Okay, so FADH2 also had some high energy electrons. Okay, and I know that's a bit hard to see here, but FADH2. Okay, and what he did was he donates his electrons to protein complex 2. And thus he now becomes FAD all over again. Okay, and maybe I shouldn't use the word donates there because, I mean, technically this thing is getting oxidized, right? Protein complex 2 is ripping them off there. And now in this case, don't forget, you actually had 2 hydrogens, right? So H plus, they both fell off of here. All right, and this is going on down here. So you have hydrogens around, right? So a lot of electrons are getting passed on and donated to the chain. So that leaves some hydrogen down here at the bottom. Fair enough. Okay. So what's the beauty of the electron transport chain? Well, when you donate your electrons to the chain, a bunch of redox reactions happen, oxidation reduction reactions. And this little mobile electron carrier, I'm going to share the videos with you afterwards so you can actually watch a little bit of a better system of how this is working dynamically. But these electrons are going to get passed along. Okay, and then they're going to get passed along some more. And this is all oxidation reduction. In other words, we reduce this guy. Okay, now he's going to reduce or oxidize him, and thus this guy gets reduced. Protein complex 3 is going to go ahead and oxidize him, thus he gets reduced. Right? And it just keeps going along. We go up here to this protein, and we come down here all the way to the bottom here of protein complex 4. All right. By the way, the electrons right here from FADH2 also join into this. So let me make this line a little darker now. What you're seeing here is the path that electrons take through the inner membrane of a mitochondrion as oxidation reduction reactions happen to drag them along, okay, or pull them along really. Okay, each one that is next is a stronger oxidizer, so he rips it away. Okay, so what you see here, this red line, is actually called the electron transport chain. Okay, so I'll write that in there. Okay, so now you can see the path that electrons take through this electron transport chain. Okay, now don't forget what I told you these electrons are. They are high energy electrons, right? These electrons right here, don't forget, they came from carbon-hydrogen bonds of glucose, right? So these things are packed. They're carbohydrate. Remember, carbs give you quick energy, a lot of energy. This is why, okay, we like these electrons. We like carbs because we can take their electrons pretty easily because we have so many enzymes to break them down. And now we give them to this electron transport chain. What in the heck is the point? Well, let's take a look here. These hydrogens that are sitting around down here, they're about to get pumped. Okay, and this chapter brings up a whole lot of chapter 5 as well with the laws of thermodynamics, because we're going from kinetic energy, we're actually going to be moving and pumping, to potential energy. All right, so we're taking the potential energy of these high energy electrons and we're going to use it to pump hydrogen. So these electrons are so high energy that this protein complex 1 right here starts to go poof, 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 and it starts to pump hydrogen. And by the time the electrons get to protein complex 3 over here, guess what they're doing is, whoops, I used the wrong color there. Let's switch back to red. They're pumping hydrogen. Wow. Right? And by the time you get to protein complex 4, I like kids to think of it this way, that you still have some energy left to these electrons. They're not quite as powerful as they once were, so you're poof, poof, poof. You're still pumping hydrogen. And you pumped so much hydrogen. My goodness, you are making this intermembrane space full of hydrogen ions, right, or protons. So these proton pumps are really, really cool. All right, tons and tons and tons and tons of this. Now, while you're sitting there watching me draw these beautiful little protons, I have a question for you. Once they're all up here now, okay, and they could be down as well depending on what direction your mitochondria is, but once you pump them all from an area of low concentration to high, what do you call all this? This is a concentration gradient, right? This is potential energy. You've turned the energy of your food into pumping proteins into a hydrogen ion concentration gradient. That's your energy right there. Whatever is not lost as heat at this point has been turned into, wow, or lost as little molecules and things like that, or some ATP. Whatever is left of your glucose just became a gradient of potential energy. Huh. We have pumped hydrogen from the matrix into the red part. So this red part, maybe we should have colored it orange to show you, oh yeah, there's a whole bunch of hydrogen in there now. Now, here's the problem though. For this to continue, we absolutely need the chain to keep being fed, but won't the electrons back up right here? Right? Well, not necessarily, because there's a very important part of cellular respiration that we started with today, and that is oxygen. And we said oxygen gets reduced to form water. The only reason that you breathe oxygen in, really, is to accept electrons at the bottom of your electron transport chain. This is crazy. So what's going to happen is, you know what oxygen is, right? Oxygen, I'm just going to draw it here. His name is O2, okay? And oxygen is a diatomic molecule. That's oxygen gas right there, and it diffuses across membrane very easily. Okay. So now what you're going to do is, technically, this O2 is going to sit over here. We call it 1/2 O2 just to make it simpler on you. I'll explain that in a second. But what's going to happen here is you have 1/2 of that O2 molecule right there, and it's going to combine with 2 H pluses. With 2 H pluses. And guess what that's going to yield is H2O. Huh. So technically, at the bottom of your electron transport chain, you have oxygen there. And oh my gosh, are we really coming full circle here? Do you remember way back when we learned about oxygen? What was one of the things about him is he's extremely electronegative. So with oxygen sitting here at the end of the chain, he's saying, hey, I have a greater affinity for the electrons than even you do. Gimme, gimme, gimme. So you have now low energy electrons. Now why are they low energy electrons here? Well, because you use the energy to pump here. Then the electrons were used here to pump this guy. They're kind of like batteries going dead, right? And by the time the electrons get here, there's almost no energy left to them because they energize this pump. So now the low energy electrons, oxygen shows up and says, hey, I'm going to oxidize you, protein complex 4. I'll take those electrons, but you can't just stick 2 electrons on oxygen, right, and think that you're happy. That would be like a neon, basically. It'd be a full outer level there. You can't have that. So then hydrogen comes in here, right? And hydrogen comes in and says, hey, let's make water. Okay, pretty cool. So you don't have to draw this, but just on a side note real quick. So O2 gas is really an oxygen double bonded to an oxygen, right? So you have 2 electrons in your s orbital, okay? And then you're going to have a whole bunch of them. Let me put this over here a little bit. So you're going to have oxygen's atomic number is 8, right? So that means you're going to have 2 in the first and 6 in the second. So let's just do 1, 2, 3, 4, 5, 6. But now this oxygen also is going to have 6 electrons. 1, 2, 3, 4, 5, 6. Oh, look at that. We just made diatomic oxygen. They're both happy. It's covalent bond. It's actually a double bond. So what's going to happen at the end of your chain is when you get those electrons, let's just consider half of an oxygen now. So again, now we're just talking about 1, 2, 3, 4, 5, 6. Oh, 2 in the first. That's not a happy. Oh, but here they come. The electrons are coming in from the chain. And now it fills you up with 2 electrons. Now you have 8. You're kind of happy. The problem is you can't just do that, right? You can't just add electrons to an atom and think that it's going to be okay. Actually, sometimes you can. But what's going to happen here is now those 2 H pluses. Remember, hydrogen has an atomic number of 1. So you really just have a proton with an electron around it. So what's going to happen is hydrogen says, hey, I don't even have any electrons right now. I'm just an ion. I'm just a proton. But I'm happy to sit down here and share. Hey, look at that. We just formed water. Okay? So technically, that's how water is made. I know it's a little confusing at the end of your chain when it says 1/2 O2. It's just talking about a whole molecule of oxygen sits at the bottom, and half of it becomes this water, and another half would become another water. That's all. Okay. So you just learned why you breathe oxygen, right? You fed your high-energy electrons to the chain, and eventually, as they get passed down, they lose their energy. Remember, if you've been following my videos, that we said that it's kind of like an energy staircase, and every time you go down the stairs, you lose a little bit of energy. That's what they're talking about here. You're losing energy in the form of pumping protons or hydrogen ions, right? But now, are you telling me that the reason we eat our food is really just to have this gradient here? No, that's not it. Okay, because what are we going to do with all this energy now in this area here where we pumped all that hydrogen? Well, we have a very special protein over here, okay, an enzyme named ATP synthase. And what's going to happen is this. Hydrogen ions here now will go from an area not low to high like we did over here, but high concentration to low concentration. All right. We are allowing hydrogen to facilitately diffuse back through the membrane. And what that does is it actually starts to spin this entire complex here. Okay. So this guy starts to spin around and around as long as there's hydrogen moving through it. Okay. And what's going to happen is, as you spin, I like to tell kids it's almost like a flower blooming, right? As long as you're spinning fast enough, the flower opens up, okay? And the bottom of this has bulbs to it. And the bulbs open up the active sites. The catalytic sites are hidden inside of here, but once you start to spin around and around and around and around and around, then you start to open up your active sites, and guess what fits in there? ADP and inorganic phosphate come in. Okay, so ADP and P come in, and I bet you I don't need to tell you what comes out. In this case, we get a whole bunch. ATP. All right, so we've been working hard to get this ATP, and we finally got a bunch of it at the ATP synthase of the electron transport chain, right? So now maybe you know why it's called oxidative phosphorylation, electron transport, chemiosmosis. This is all redox. You know, he's going to oxidize him, and he's going to oxidize him, and he's going to oxidize him, right? okay, thus getting reduced the whole time. And now what did you do? Well, okay, the final electron acceptor is oxygen, so he got reduced to form water. So oxygen oxidizes this protein, gets reduced to form water. He's out of here now. That's a waste product, right? So if you remember back when we started, we said, oh, yeah. Okay, oxygen gets reduced to form water. And the whole chapter, we've been ripping away the electrons and releasing CO2. Now we finally got to the energy part. There we go. Because you have all this energy available up here as potential energy, right? So you can go ahead and allow it to diffuse back down through this special enzyme. As it opens up catalytic sites. There we go, right? So this whole chapter is almost just energy coupling, right? You're changing energy from one form to another. You started off with beautiful glucose, and you just keep moving through to a different form, different form, different form, different form, and finally, we're where we want to be, okay? And of course, heat. There's heat all over the place here, right? Lots of heat energy. So this is no joke, right? Your mitochondria is your little furnace, right, of your body, of your cells for sure. So pretty crazy stuff here. Okay, so let's talk a little bit more about what's happening here. First of all, this, I'm sure you could already tell, was the electron transport chain. Right, and then over here, I don't love this word, but this is what we call it, chemiosmosis. Right, so there's our two parts to this. And what do you think it's called? Okay, let's make a little key for ourselves here. When you are pumping from an area, now notice I'm using the word pumping. When you're going from an area of low concentration to high, oh baby, what do you think these red arrows represent then would be? If you're good, remember transport? Active transport. Right? And why is it active transport? Again, you're using energy and you're going from a low concentration to a high concentration. Right? So, using energy to do, well, where did you get the energy from? I don't see, Mr. Boylan, there's no ATP being used over here. Ah, the electrons. Right? The electrons gave you the energy for this chain to work. Now, what do you think the blue arrow represents? Right? In this case, you're going from an area of high concentration to low. It's like a tennis ball rolling down a hill. That's easy to do, right? That would be passive transport. And like we said, because you're moving through a protein, this is like diffusion, right? Or facilitated diffusion across membrane. So in this case, you're going from a high concentration to a low concentration. Now, what I usually do here when I'm in class is I like to ask the kids, well, why are you always going to have more hydrogen up here than you do down here? And there's a couple of reasons for that. The most obvious reason is, well, you have 3 pumps up and 1 facilitated diffusion going down. So that's your main reason, right? Another reason that you'll have more up top, look what you're doing here. You're always stealing 2 H's for water. Another reason that the concentration will always be higher here, and it doesn't have to be on top, it could be down here wherever it's red. Look at the small space. It's much easier to get a high concentration, okay, into a tiny area than it is into the huge swimming pool in here of the matrix. All right, so it's a confined space. But you're not going to run out of hydrogen down here because, oh yeah, we're always delivering them, right? You're always delivering, the NADHs are always dropping off 1, the FADH2s are always dropping off 2. Right, so you'll always have hydrogen available. You're always going to have some hydrogen spilling back through. Right? Okay, let's talk about our totals. Now, these are, they're in a state of flux all the time. Some scientists like to talk about changing these up regularly. And a lot of it deals with like shuttling the NADH from the cytoplasm into the mitochondria and how much energy you can actually get to pump hydrogen. But for our part, it doesn't really matter again. So we're going to say, in this case, you got about 28 ATP. Pretty cool. All right, so we got 28 ATP. That's pretty cool. And in this case, I also like to think that we also got some NAD plus and some FAD. Oh yeah, don't forget, that's going back, so we can recycle these guys. So I like to think of the NAD plus and the FAD as a total here, because yeah, we're going to go reuse them again now. And of course, like we drew up there, heat. We lost a lot of heat in the process. As a matter of fact, what's more efficient, right? A beautiful pickup truck driving down the street, okay, which was made by man, and you know, some of them are very nice, or your body, which has had billions of years, okay, of evolution since life began. So what's more efficient? Okay, and the answer would be, well, for the most part, the truck, right? Because you lose so much energy. I mean, so does a car. You ever put your hand on an engine? I hope not. That's been running because it's really hot. But boy, oh boy, you lose a ton. You only got 28 ATP, and we'll talk about your grand totals in a second, but 28 ATP from this step. Well, let's do that. Let's see your overall totals then. So let's just talk about your ATP. So overall ATP totals. All right, here we go. How many did we get from glycolysis? If you remember, we got 2 net. Remember, we used 2 to start, and we gained 4, so we call that 2 net from glycolysis. Okay, how many did we get from the grooming of pyruvate? Was 0. So let's skip that. Now let's go to the citric acid cycle, or the Krebs cycle. If you remember, we got 2 ATP there. And now how many did we just get from oxidative phosphorylation? Well, we got about 28. Okay, so about 28 ATP. So technically, how many ATP do we get altogether? About 32 ATP altogether. Okay, the thing is, that's only about, okay, about 34% of glucose's energy. All right, so what does that mean to us? Well, it means that your overall ATP totals, we got some from glycolysis, some from Krebs, and then a whole bunch from oxidative phosphorylation. But that 32 number there, okay, that's only 34% of the entire energy that we possibly could have gotten out of glucose. And you might say to yourself, wow, we're really not efficient? You'd say, yeah, you're right, okay? And that's why we're always breathing oxygen, we're always eating. We are dirty machines. We're basically furnaces, right? So what if you had a friend that was born with a genetic disorder and they were 100% efficient? Well, you wouldn't have that friend, right? Because he or she would be dead, okay? Because we now need this. Our bodies are designed to lose all of this heat. As we went through our drawings today, we lost so much heat, and that's important because that's where we work best now. Our enzymes work at that optimal temperature. Okay, so pretty neat. So what did we learn today? Oh my goodness, we did a lot, right? And if you stuck with me through all that, I know that took forever, good for you. You're an all-star. So we started off talking about the whole point of cellular respiration, okay? And it's just redox reactions. Okay, you oxidize glucose this chapter, and yeah, you release your CO2. What did you do with the electrons that you oxidized here? You took them out, you gave them the oxygen at the end of the chain that became water. Why did we do all this? To get ATP. Now you started off in the cytoplasm, okay, and that's through glycolysis. We only drew the carbons today. But to start breaking down and getting energy out of glucose, you needed to invest 2 ATP, all right? So the 2 ATP went in, and now you can phosphorylate this molecule, you can bust it apart, and now you get some high energy electrons, right? And some hydrogen is released. That's thanks to dehydrogenase enzymes. And yes, now in this energy payoff phase, not only did we get these high energy electrons, we also got ATPs. Great. But the majority of our energy is still here in pyruvate. Now, how did we get ATP this chapter? Not talking about the very last step there, but anywhere else, we're just talking about like substrate level phosphorylation. Okay. Now, what's going to happen is, now that we have our pyruvate, well, we're not necessarily ready to roll yet. We're going to have to go ahead and groom that pyruvate because we really want to get to the Krebs cycle. Pyruvate is allowed to diffuse into the matrix of the mitochondrion, and now you're going to get it ripped apart, okay, a little bit, steal some electrons, and you're going to have this coenzyme A come in and pick you up. you're now acetyl-CoA. Now, where can the acetyl-CoA go? Well, coenzyme A says, hey, come on in here. You're still in the matrix. I'm going to introduce you to this person named oxaloacetate. So the 2-carbon acetyl group mixes with oxaloacetate to form citrate. And now, poof, you just get destroyed, right? And what's left over is oxaloacetate, so you can start it all over again. But the whole point of this was really to oxidize what remains of glucose so you can steal those electrons. Why so? Well, we want to deliver them to the electron transport chain. So inside the mitochondrion, along the cristae, okay, the folds of the inner mitochondrial membrane. And remember, why in the world are there so many folds? Well, think about all that beautiful surface area, right? The more twists and folds and things like that you have, the more of these chains you can make, right? And all these chains are going, and you deliver your electrons, and as they get passed along, right, so you reduce the chain, NADH and FADH2 reduce the chain, or they get oxidized. Now the electrons get passed along, so you can pump, 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 right? And then oxygen comes in, takes the low energy electrons away, and forms water. What did you do with all of this energy, right? Because you have potential energy in a hydrogen ion concentration gradient now. You allow it to diffuse back down through ATP synthase. That opens up the active sites of the enzyme, so ADP and P can fit in, and whammo, you get ATP. And from 1 molecule of glucose, we get about 28 ATP at this step, and that means total, we got 32 ATP, right? Pretty cool. If you were able to stick with me today, that's amazing, and I hope these drawings really help you out. Okay, take care, Beaklings.